So with that, I'm going to hand things over to April. April, thank you so much for helping us with this and facilitating this. I really appreciate all the work that your team has put in.
Thank you, Liz. I hope everyone can hear me. I think Susie is going to share the slide deck in a moment. My name is April Clyburne-Sherin, and I've been involved with this initiative of a community-led reproducibility education project for the last few years, and I'm really excited to join you guys today and talk a little bit about reproducibility. So next slide. So my background is epidemiology, and I currently work in education and reproducibility. I'll do the introduction today. Next slide, please. I want to start by just saying thank you to some of our sponsors and also to the societies that helped put this event series together. They've been really helpful, and we've been really excited about doing this webinar, so appreciated that invitation. Also, I'd like to send a special thank you to Benjamin Schwessinger, who has been one of the originators of a lot of the content that you'll see today, although it has evolved over time, and you guys know all about evolution. So the current state of it has been the work of many, many volunteer contributors. Next slide. So as Liz said, you can use the built-in features to ask questions along the way, and we're going to have time at the end to answer all your questions. But I'd also encourage those of you that are able with your tech setups to also join the shared notes documents. The link I just added to the chat there a few moments ago. And this is a place where you can ask questions, but you can also share your experiences as we move through the content, because we have a lot of content and it's going to get a lot of you thinking about things that have worked for yourself in terms of improving the rigor and reproducibility of our work. And we encourage you to share those experiences in the shared notes doc. And feel free to add a +1 to those questions and comments that you find most helpful. Next slide. Great, so let's get started and sort of introduce what we're going to talk about here today. So why does reproducibility matter, and why does it matter to you? Next slide. So many of you filled out our pre-workshop survey, and so this sort of leads to, I think, the main motivation for all of us in being interested in these topics, and that's our own personal experience with trying to either reproduce our own work a few weeks, months, or years down the road, or trying to reproduce somebody else's work, either a colleague in your lab or trying to build upon the experiment that someone else has published. So it's a very common experience for people to have a hard time reproducing their own research. But it's an extremely common experience for people to have a hard time reproducing someone else's research. And many of us, especially as we go through our careers over many years, have had problems reproducing both our work and someone else's. So this is the core focus of like what motivates us to try out some of the things we're going to talk about here. Next slide. Oh, you already did. Okay. So the goal of these webinars is to introduce people to reproducibility. So give you just an introductory framework so you can start to kind of explore these ideas, including what reproducibility means, what the different facets are, and that sort of thing. We're also going to focus on some of the practical things that we can do as individuals, the tools and the methods that we could start to bring into our own work to advance reproducibility and nudge it down what we call the reproducibility spectrum. And the idea of a spectrum is important because reproducibility, it's not an all or nothing state. It's a lifelong project of learning. So all of us are going to, take some time to reflect on what we can do with our own research, but over time we're going to learn a little bit more about what works and what doesn't work for improving the reproducibility of the type of work that we do, as well as the landscape of reproducibility will change. So there will be new tools available, new methods available all the time. So think of this as the starting point of a lifelong journey. So what does reproducibility mean? We'll talk about a little bit about that. We'll talk about the different types or modes of reproducibility and how they relate to one another. We'll talk about how reproducibility fits into other issues that we should reflect on as scientists, and how our work should be shared, how we should relate to other scientists' work, and how we can improve the efficiency of our own work. And then finally, we're going to dive into the reproducibility tool shed. So this is, throughout this whole webinar, we will introduce tools and methods that somebody in our Reproducibility for Everyone community recommends. They've tried it out for themselves, and they feel like this will help many researchers to improve reproducibility. And the 4 facets of reproducibility that we are going to focus on are improving the organization of your research, improving the documentation of your research, automating the analysis of your research, and disseminating or sharing your research. So these are the 4 main facets of improving reproducibility. Next slide. Okay, so what are we even talking about? What does reproducibility mean? So reproducibility actually is not a universally decided definition across disciplines yet. So we're going to introduce 2 of the very commonly used ways of defining reproducibility that really can bring you into diving into what reproducibility means in your discipline. So to get started, we'll talk about the base level of reproducibility, which is reproducible research, also sometimes called computational reproducibility. So reproducible research is when an author, scientist, or researcher provides all of the information needed, all the data, all the analysis code, all the methods that are needed to rerun that same analysis on that same data and that same code, and to get the same results. So the same tables, the same numbers, the same figures that they originally shared, say in a paper. So this is baseline reproducibility, just rerunning the code and the data and getting the same results. And then a little bit more advanced than that is what people call replication. Replication is when you're doing more than just rerunning the same data and the same code to get the same figures. You actually want to rerun a study using the same methods, but collecting new data and running new analysis. And so this is a little bit more advanced. You are trying to see whether or not the study can actually be rerun and replicated. Next slide. So there are different types of reproducibility, different levels you can think of. And so I'll bring out the idea of a spectrum again. This is, these ideas sort of build upon each other. You see there in the first box, reproducibility is, again, using the same data and the same method. So the same analysis code, the same statistical tests, that sort of thing, on the same data and getting the same findings, the same numbers and figures. If you're able to analyze somebody's data with slightly different methods, maybe using a different statistical tool, using a different statistical test that's getting at the same sort of relationship in the data, then what you say is that that research is robust. So it is not only reproducible, it is also robust. So you're moving down that spectrum. If you're able, oh, sorry, could you go back? Thanks. If you're able to collect new data by running a new study and use the same methods, that's what we said before about is replication. And then finally, if you're able to use different tools, different methods to analyze new data on the same relationship, then we have generalizability. So these are related topics, and what we're really aiming for is that generalizability over time across our shared knowledge. Next. So is reproducibility all that matters? Next. Reproducibility is just one of the parts of scientific rigor that we all are trying to work towards improving. So we're not touching upon all these other issues today, but they are related, and reproducibility is just one part. Next. So one thing to keep in mind, especially for those of you that are early career, is that there are many sort of resources out there that will tell you, how to improve, the logic of your study design, how to properly use statistics to analyze your data, all of these different parts of rigor. And the thing to keep in mind is that this is a lifelong learning process for all scientists. No one is perfect, and we've all done studies in the past that we would do differently in the future. And don't worry about adopting all of the best practices with every project. Just start with one little bit. Every little bit helps. Everybody needs to start somewhere. So if this is all new to you, that is totally okay. Take what you think you can do now and build on it in the future. And you'll move towards creating more transparent, more open, more reproducible science. Next. So we're not going to focus too much on the negative side of reproducibility. There are plenty of papers out there that talk about the factors that decrease reproducibility, that focus on all the things that are going wrong in science. Next. And these range from the technical factors to the human factors to problems with how we reward scientists, the career paths we've created for them, as well as how statistics are really applied in the real world. Next. There are many of these factors that we could dive into. So next ahead a couple times. But today we're going to focus on the positive side of things. We're going to try and focus on those things that you can do to counter some of these. So there is a body of literature if you're interested in this stuff, but we're going to focus on the positive, what we can do, the practical things that we can do to improve our own work and to make our lives a little easier. Next. Our perspective is one of optimism. We understand a lot more now about how science and research is done than we did in the past. And because of that, we can use that information to do better and to accelerate science. This scientist, Corey Bargman, she's an HHMI investigator and a president of the Chan Zuckerberg Initiative. She made a great quote that 82 years ago, there were no antibiotics and we didn't know that smoking causes lung cancer. When you first learn about these things, you can really accelerate, science very quickly. So we can expect the next 82 years of scientific research will do a lot to improve society, to advance society. So we want to focus on the fact that what we're doing right now as researchers is working. We are improving knowledge, we're advancing knowledge. And when you start to reflect on reproducibility, what it allows you to do is accelerate that building of knowledge to make it more efficient. Next. So with that in mind, where could we be in 82 years in scientific research if we all work together to accelerate science, to do more, to share more? Next. So many of you filled out this survey in advance of your greatest potential for growth in terms of adopting a new reproducible practice. Next. And many of you agreed that sharing more detailed methods, better descriptions was your primary way of improving reproducibility. So we'll talk a little bit about that. We'll also talk about data sharing, which 83% of you said was a major improvement that could help, as well as better code sharing. We'll touch upon that as well. The other main ones were less pressure to publish significant findings, increased power, and reagent sharing. So we'll talk a lot about the ways that you can improve your research in these practical ways. Next. So again, there's going to be a lot of information in the next hour and a half, 2 hours, but focus on just getting started. Take one thing and get started to improve the reproducibility of your work. Just doing one thing will help. And focus more on what you can do. Don't worry about what you can't do, because no one is perfect. Next. So adopting just a few of these isn't just good for you, it will actually save you time in the long run and save you from disaster. If you, instead of losing your work, instead of forgetting what you did, you'll be able to know you've got that ready. It's reproducible for you, it's reproducible for everybody else. Next. Great, thank you.
So, hello everyone, my name is Susann Auer, you can call me Susie. I'm a plant researcher and a lecturer based in Germany, and I'm working at a German university. You can find me on Twitter. Here's my handle, I will pop up again in the end at some point. And I want to lead you a little bit through the data management part. So now we jump right into the practical things, what you can actually do to implement to help make your research more reproducible. So data management is one of the most, I think, crucial tools we have at our hand, and it's a good thing to start with data management when you want to improve your research. So you maybe are familiar with some of these findings when you've done a study, you collected some data, and then maybe you go on vacation or you do something else, and then you come back and you want to do that analysis, and then you're like, oh, what did I do actually? So where's that file that I had then? Or maybe if you just look at the file names here in the middle, you see that you probably did not give it a good file name, or you lost that one very crucial information. Was it the wild type, or was it the mutant strain that I was analyzing? So maybe you are familiar with some of these frustrations that then arise. So what we can do is actually make a data management plan. A lot of funders nowadays already request to make a data management plan, and there's a nice tool for that, the data management plan tool. So if you look at their website, they have plenty of resources to help you make a data management plan for whatever project you're tackling. However, if it's not requested, or you're working on your PhD or something and no one forced you to do that, it's also something you could try to come up with. So maybe get some inspiration here at the DMP tool. And then we emphasize it all the time, you don't need to become a superhero overnight, but it really helps to have a data management plan and then be happy, because that's what counts at the end of the day. So what you see here is the life cycle of data, going over from planning, over collecting, assuring, describing, and so on and so forth. You can read that yourself. So the first thing to think about when you are starting a project is, what kind of data will be produced as a part of my project? Am I in the field collecting data and I just have some handwritten notes, or is there a machine that I get data from? What kind of stuff do I actually have that I'm working with? I hope you don't see the messages popping up on my screen, which are of course popping up right when I give the talk. I'm sorry for that if you hear them. So what data will be produced as part of the project? And if you have identified all the types of data that you will produce, then you can sort out, how will I organize these data? So that's what a data management plan does. It states specifically what to do with what, who's going to do that, and so on and so forth. So how will you organize your data? Will you work with spreadsheets? Will you work with some statistics software? Will you have some piece of code analyzing something or organizing it? How will you document your data? How will you standardize them? How will you store them? I will come back to the storing part, because this is really crucial. How will you protect it? Probably if you work for a company or something at some point, then you need to ensure that the data you created are not available for other people who don't have the right to see them. And then, of course, if you collaborate with other people, or if you have your supervisor or you have a student to share data with, how will you do that? So they can read the formats you have your data in. How will you archive your data when the project's finished or you're not working on them? Super crucial, specifically in collaborative projects, is to decide who will take responsibility for carrying of any of these activities I just described. And then that's also something a data management plan states. When will these activities take place? So when you're working with someone collaboratively, you're the person responsible for collecting the data. And then you hand these over to someone else, and that person should know what his or her role is then, if they are analyzing them or whatever they are doing with them. So the responsibility part, this is really a big sanity saver, and it should be discussed before actually working on the project in the best stage. And then very crucial, sometimes overlooked, are metadata. So metadata are the data describing the data that you have. They provide the context or the field. Maybe they can describe something if you are working in the field, and then you have to, like the weather is a big part of your data collection, and of course you have to take down what was the weather or what was the environment, or metadata could also be you work in a greenhouse and then greenhouse was failing and like your plants were sitting there for 3 days, not seeing any light. Of course, that would be also a big factor that impacts your whole experiment. So metadata in any case are data about your data, and they should be collected and stored accordingly. You could make a README kind of file if you want. So we always encourage people to actually develop when they store their data. So you have your kind of structure and you thought about how you're going to keep your data, to have something like an informative directory structure on your computer. So for example, it makes sense to keep research materials together for each part of the project that you work on. So here we have an example, project 1, and then you would have your folders, some subfolders. You will have a methods folder where you put all the PDFs of your methods, for example, or the links to protocols or whatever you are using. We're going to visit that again later. Then you ideally have really one folder for where you keep your raw data, and these are going to be separate from your analysis data. And then you just see, these are just some examples how you could make a directory structure that makes sense. To have some scripts in one folder, then you have a manuscript, your first ideas of writing up whatever you have seen, what you have analyzed, and then you have a README or you have a link to an electronic lab notebook. So this is just some inspiration for you how you could organize your folder. Very important, this is why we write it out here so specifically. Really try to keep the raw data at one specific place and do not touch it all the time again. So either you make a copy when you do an analysis, or you're going to find another way of not touching it and not accidentally doing something with it that changes your raw data. So really try to keep them separate. A second thing is to always back up your data. I think it's Murphy's Law that whenever you're at the end of your PhD, something's going to happen to your data. Your computer is, I don't know, you spill some tea over it or something happens to your hard drive. So it's always best to really back up your data specifically at the end of projects when you've basically done everything to collect and analyze your data. It's best to have 3 separate places to keep your data, so 3 unique locations really. Probably have the cloud service of your institution, then you have another server where your data are stored, and then you have a personal drive. If you don't have in your own server, or there's no server from your university available, then have 2 personal drives, for example, and then you store one at your workplace and one at home or something, so, or at your parents, whatever you want. So just find ways to make this happen. It's a big sanity saver, and trust me, I've been there as well. So then you have that nice directory structure. Now let's just quickly jump into what are useful file names, what are useful names you could name your files. So this comes back to the frustration we had earlier. What did I call my last file? So this is what that cartoon here is summarizing. I really advise against ever naming your file final. It's better to work with versions, to say, okay, so this is like analysis 5, and then it's version 1, 2, 3, or whatever. The good thing is, and it's a big sanity saver as well, is to have some rules. So a file naming convention are basically suggestions of what you can name your files. So it makes sense, for example, to always include the data in a year, month, day format. to use meaningful abbreviations that actually are meaningful to you and to others. So standard abbreviations from your field are totally fine. Abbreviations that only you came up with are probably not easy to understand for others if you think about sharing your data. So maybe try to keep it in a way that's also understandable for others and that you will also remember in 5 months or so from now. Have some group identifiers. Then if you change your style of naming something, you should document your decisions, why you did that or why you decided for this so it works better. Try to be consistent and use version numbers. So these are, as I said, just some suggestions. If you Google file naming conventions, you will find a lot of useful information that's out there, and some systems work more for other people and others work more for another group of people. So the rules themselves do not matter so much. It matters that you have rules in place. This is an example for a very specific file name. So you see here, it starts with general, and you see that on the left side, and then it goes until the specific part. So you have a date, you have a project identifier, an experiment, the type, so this is data, it could also be the manuscript or something. Then you have an idea. That's really for collaborative projects very useful to have an ID, whose file is that, who was working on that, or who created it. And then you have a version. So this is a useful file name. I know it's pretty long, and the reason why you have these underscores or dashes is to make it easier for people who run code, but you probably know that. So if you're collaborating with people who do a lot of coding or who do your analysis, if you're working with bioinformaticians, then it makes a lot of sense to not have any spaces within the file name, because that makes it a pain for the person who's doing the analysis to actually import these files into their programs. So the second thing I want to touch on briefly is ELNs, electronic lab notebooks. So paper lab notebooks are in place for a very long time. And you know probably the originator of this one on the right, Leonardo da Vinci. They are still very common in they are very commonly used still. And I had actually a question to one of my seminars when I was presenting that slide. Someone came and said, I don't need an electronic lab notebook because, I mean, Leonardo da Vinci's lab notebook is still available. We can still read it. If that concern comes up, or if people say, well, it's not worth thinking about electronic lab notebooks, I just want to briefly slip in here that, of course, we can still read it, although it's from the 15th century because someone preserved it, right? So paper usually would not withstand hundreds of centuries because of, you know, getting moist, burning, or being dislocated, or other kind of stuff. So the negative thing with these kind of paper notebooks is they are hard to share. If you're collaborating with another lab, then your copy is here, and then they don't have a copy of what you are working on. So you cannot really search them unless you are good with having a content list in them. They can be damaged, misplaced. They are not easy to back up. You have to, I don't know, scan them or something. And so these are some disadvantages with paper lab notebooks. However, they are still being used, and this is not a bad thing. It's just maybe an idea to think a bit in terms of electronic lab notebooks, what we could probably gain from using these. So an electronic lab notebook, and I come to what specifically they are in a second, they are searchable because it's a digital file. You can export data, so you have written up your method, for example. You can easily export that into a PDF and share it with your collaborators or your PI or your students. You can basically generally share these easily with other people. You can embed high resolution images, protocols, you name it. Whatever you have as digital data you can share, or some videos or something. If you're changing labs or you are working on another lab in a different location for some time, you can have access globally, or if you're now working from home, you could have still access to that electronic lab notebook, and some of them also come with a mobile app, and then you can quickly upload pictures or whatever using an iPad, for example. So there are a few basic features that all of these electronic lab notebooks have, and these are very often that you have, so you see on the left here, you have some sort of a drop-down menu when you see all of these files that you have started and saved. You will have a search button, you will have some Office compatibility depending on what lab notebook you use, but they are pretty open right now, so they usually you can export something into a Word document, for example, or PDF or something. You can print, you can share, you can have an attachment there, so like this is a jail picture. And then of course you can create folders for your experimental data or for ideas you have, and all kind of stuff that you would usually record in your normal lab notebook. And then of course the nice thing is that each of these entries are dated, so you know specifically when you've been working on these. So we have, these are just some general options. There are options of electronic notebooks that are paid for. So ideally, your institution will purchase these, they will decide for one lab notebook for one company basically, and then purchase them. So these are the paid for ones, and then you can use them. Then there are some paid for ones that have a free version, for example, Zynode or Benchling, which a lot of people are using. So it's always good, that's my advice actually, to first try it out, how you can, if you can work okay with that electronic lab notebook, or if this is something that you don't like, and then you maybe try out another one. So just give it a chance, I would say. Then there are, of course, open source versions. So we're coming to a few more options. So these are really not the only options out there. And then there are the free versions. So you can use the Open Science framework, of course, as an electronic lab notebook or local wikis. If you visit that data page, that website here, data management, and then at Harvard, you're going to access an internet page, which leads you then, if you click on that thumbnail picture that looks like this will lead you to a Google Doc that's updated very regularly. So I think the last update was something like June 2020. So this is pretty much up to date. And what they do is they just tell you what kind of electronic lab notebooks are available, what parameters you could consider. So even if you don't know what you're looking for right now, you can just look at that huge Google Doc and then just see in that decision matrix, so to say, what you would give a chance and maybe try out. And this is really, one size does not fit all. It really depends on what field you are in, what you're actually doing. So for example, a chemist person who has to draw chemical structures maybe goes for that specific lab notebook because that's one of the strengths of this one lab notebook. And then another person like a biologist probably goes for a completely different one. So you really have to try them out. Some very general tips for electronic record keeping are, if you do it with an electronic lab notebook or not, you should back up your data regularly. Weekly is like the best thing. Then nowadays, still maintain a physical observation notebook in parallel, just to make sure that everything's, in case, I don't know, the server fails or something, that you still do not lose all your data. This will be less of a concern, I think, in the very near future. And then, of course, if this is important to you, you can check if they have some mobile apps. And this is, the last point is very important. So if you use a free electronic lab notebook, usually they will store your data on their own servers. And if you're for example, because I'm not in the US, so if I have my data stored on a US server, that will get me into trouble if I'm working for a European company or something because of data protection rights and laws and that kind of stuff. So you need to check data ownership policies, really. Usually the paid for versions allow you the access so you can store the data on your own server and maybe have some sort of an offline app or something. Back to April.
Thank you, Susie. Next. So we talked a little bit about how to improve the documentation of our work through better organizing, and using electronic lab notebooks. So let's talk a little bit about then sharing our methods. Next. So one of the problems we see in the real world when we share our methods is that we actually are missing a lot of methods information. And so this is a common thing that we encounter. We see on the right this Twitter post where someone was looking for a protocol or a method that was needed for a study. And it was referring to a description of that protocol in another study. So the researcher would go look in that study and they would find, again, a citation for this reference being from another study, and you keep going back and then you hit a paywall or you can't find that study. So oftentimes we can't even find the methods that we're trying to replicate. Next. Another thing that often happens is that we do find some information about the methods, but it's not actually enough information or detailed enough information to actually reproduce them for ourselves. So on the right, we see this Twitter post. Another very common thing that we've all encountered is we see a method that is referenced in a previous paper. We go back to that paper. It's referenced in a previous paper. We go back to that paper. It's referenced in a previous paper. We get to the original paper and it says, devices were fabricated with conventional methods. So you're kind of left shrugging your shoulders, not quite sure exactly how that work was done. Next. So one sort of satirical take on this is described here. If we used these sort of processes that we often encounter in science when we're trying to reuse each other's methods, if that had been, say, a drawing tutorial, in this case, to draw an owl, it would be the equivalent of step 1 in how to draw an owl is to draw some circles, and step 2 is to draw the rest of the owl. So there's clearly an issue here where we are describing methods in ways currently that make it really difficult to reproduce someone else's work. In this joke, they describe how the owl was drawn by describing the paper that was used, right? So the size of the paper, what weight of the paper was, what the brand of the pencils were, and then it just says, we did so by looking at owls and drawing what we saw on paper. The protocol yielded one drawn owl. So I'm sure this sounds familiar to many of you that have struggled in the lab to recreate something that you read in a paper. So what should we do next? What we want to do is to share detailed methods and share them in public, easily accessible ways. So we want to be using what are called methods repositories or protocol repositories. We don't want to always be relying on adding supplemental files to our publications because that will make them only easily discoverable to other people that are looking at our paper and to people in our discipline who are reading that particular journal. Next. So 2 examples of places where you can publicly share your protocols. One is Protocol Exchange and one is protocols.io, and these are both free to use. Next. One of the other advantages to using a repository to share our methods is that you can actually have researchers across disciplines working and improving upon the same method. So this is an example in the real world of someone who is looking for a particular RNA extraction method, and they were using it for their study on primary cortical neuron cultures. Next. So one particular person knew of a public protocol in a repository and pointed them to it. Next. And this particular protocol, it was published in a discipline-specific part of a journal that was referring to a stickleback parasite. So this particular researcher who was working in one field wouldn't necessarily be familiar with and be able to discover easily that particular method because it was working on a different experimental system. Next. So when we share our methods in our repositories, we make them more discoverable, we make them more reproducible, we can facilitate more collaborations and connections across disciplines, we make it easier for other people to reuse our work, and it can also increase the value of our research because we're creating this sort of additional publication into the scientific world in addition to just our paper. Next. So let's take a stab and see how difficult it is to follow a protocol and to write a detailed protocol. So we'll take a step back from this sort of fire hose of information that we're sending to you right now. See if you have some paper and a pencil, grab that. If you don't, then you can open any sort of drawing software that you might have on your local computer. So I'll give you guys a minute to locate that if you want to participate in this short exercise. While we're waiting for that, I just want to give a shout out to those of you that are sharing your questions and also sharing answers in the shared notes doc or in the Q&A, because I think that's one of the great values we can get out of these webinars is by sharing each other's experiences. So I'll give one more minute for people to grab a pencil. Okay, next. All right, so we've got a protocol here to draw a picture. So we're going to start by drawing a small 1/8 of an oval, so not a full oval, in the center of your paper, with the open part facing to the bottom left. Then we're going to draw 1/3 of a large circle underneath the oval and draw small perpendicular lines on each end of it. At the center of the 1/3 of a circle, we're going to draw 2 squares with a small gap between them. Then we're going to draw a wavy line under the squares. Step 5 is on the ends of the larger circle, draw smaller half circles around them with 3 dots in the middle. Now draw 2 big circles on each side of the small oval from the beginning, and they should be more on the top than on the bottom. In the big circles, draw smaller circles, and in the smaller circles, draw even smaller filled-in circles. Now draw a wavy square around everything you drew so far, and then draw 7 small ovals within the space of the square. Okay, for those of you that followed along with that protocol of a drawing, any guesses at what it is that you just drew? Anyone want to put into the chat what they think that might have been? Any guesses? Let's see. Mickey Mouse? It's a good question. A weird frog. Yeah, could be. Next. So what we have here is a good example of how when we write down our steps, the things that we do, it's actually very difficult to write them in a detailed enough way to get the same results when someone else tries to follow it. So we've run this particular protocol drawing exercise, and these examples that we see on the slide are some of the best versions of it, because when I first drew it, a weird frog is probably pretty close to what I got. Next. And what the original drawing was as a drawing of a cartoon. It looks a lot like SpongeBob SquarePants. So this is just a sort of a quick sort of example of, like, we know what we're saying, sharing detailed methods about what you do in your research. We know it's difficult to do in a reproducible way. So we should spend some time sort of practicing these skills of improving our protocols, improving our documentation every time, getting a little bit better at it. Next. So we would definitely be able to accelerate our research if we started to think about the sharing of our methods and our protocols as scientific publications that have value, that we should practice, that we should learn to improve, that we should iterate on over time. So think of a protocol as a self-contained scientific publication that others can reuse. Include an abstract that puts your protocol in context so people can understand, and then add as much detail as possible. Think about the duration and the time for every step. Think about the amount of the materials that you use. Be specific about the vendors and the the names of the tools that you're using. Refer to catalog numbers specifically when possible. With every step, describe the expected results so people that are trying to reuse your protocol will get a sense about whether they're actually doing it right or not. Include safety information, include all the software and the software packages that you rely on. Make sure that you put the steps in order, because sometimes the order you do things in is important. Anytime there's a specific tip or trick, add that into the protocol. And then use versioning so that as you continue to improve and update your methods and your protocol, everybody can always access the most recent, most up-to-date, and best version of that protocol. Next. Oh, okay, yeah. So in addition to sharing our protocols, we also want to think about sharing our materials, because this is also part of our methods. So just very briefly, when you're using reagents, you want to record as part of your methods how you created them. You want to be sure that you authenticate your reagents so that you know that you're using what you think you're using. Provide references to all the protocols needed to use it properly. Use standard naming conventions when you come up with a name for your reagents. And use a RRID. Just 1 second, I have to look up for myself. An RRID is a Research Resource Identifier. You can get one of those for free for your reagent if you've created it yourself, or you can reference that if it's been created by someone else, or you can use your reagent's catalog number so that people know that you're referring to the same reagent. And then finally, they also have repositories for sharing reagents. So take a look into that if you are using reagents in your research to make it easier for other people to reuse your work. Great, thank you.
Hi, my name is Małgorzata Gazda, and I use my nickname Gosia. And I'm evolutionary biologist. I finished my PhD at CBO University of Porto, and I just started my postdoc at ENS in Paris. Next. And I would like to walk you through a little bit through like getting reproducibility applied to your bioinformatics, to your computations. Next. So here we have the same problem as we already know, that everyone basically faced problems reproducing either own or someone else's work. And this, well, surprisingly or not, applies also to computer science. Next. So I think this can be a quite common feeling, like, what program did I use? What comment exactly did I use? Like, what were the specification, the details I used? Where did I put my data? I want to reanalyze, so I try to open the same package. The package doesn't exist anymore, or the version of the program was updated, and then the package doesn't work in the new version. Oof. So the most common problems will be the problem with the dependencies, and the other problem, if you don't like make a detailed notes in README file, will be like, how did I exactly do that? Next. So then it is very important, the same like for the lab, to actually document your programming and make a very detailed documentation, what, how, and why exactly did you do that. And this will help you not to face the problems like, or not to look for the same problems and solutions 2 weeks or 6 months later. And it will help you also to share the ideas and then the ways to do the analysis with your colleagues, your collaborators. So the best approach to do that will be to put like all the documentation in one executable document. And then there are some attempts to try to put like interactive version of the paper that you can actually reproduce the analysis reading the paper, and that's quite interesting idea actually, I think. And applying all this makes it that you can easily share whatever analysis you did. And to start like making your own protocols for data analysis, basically I would recommend looking at Jupyter notebooks or the RStudio. Next. And here you can see the example of Jupyter notebook. So you can see that there is explanation of the idea in the top. So like you write down what kind of problem you want to solve, why and how, and that will be very beneficial for writing later on the part of methods and discussion in your future publication. And then on the bottom right part, you can see the exactly code that you wrote. And on the left side, you can see like how the code is executed and what is the result of that. And that is really, really useful. So in case there's any mistakes, you can easily find them. Next. And then the problem that Susie already discussed, like having the naming of the file issue. This happens also with the document changes for the code. So a new approach to face this is the version control system. And this is either done by Git or GitHub. And then GitHub has like the social media component, so you can discuss the things and share with others. So in this way, you control the version of the document. So what was done, when was done, and who done it. And then, well, GitHub basically let you share your code easily and collaborate on your code more easily. Next. And then also quite common problem working on the server is that how to install the different packages, and sometimes they would not work, sometimes they will not be available, and then one of the files, dependency files, is missing, so then you cannot get the final package working. And then other versions can be conflicted, so you will never make it to work. So what is the solution of this? Next. This is implemented quite easily, actually. So you can use the environment, which is called Conda or BioConda. And this will enable you, for example, you want to use one software. So then you kind of going through the conda, you will install that software, kind of read the software to your working environments, but not only that software, but also all the other softwares, so dependencies that you need to execute it. And then this will basically free you from the dependencies hell and versions conflict and so on, because you just read in whatever is there, everything is updated and everything runs smoothly. And the similar thing can be also apply using our projects in R. Next. The other way how to cope with this is to make your environment portable with containers. So the container will be like, imagine like a box that has everything that is needed to basically replicate your analysis. And this box is called an image. And this image can have like everything what you need. So the code, the programs, the environment, and then no installation is required for anything. And then you can just spin an image into the container using the Docker or Singularity. So this will be like sharing basically your computer. Next. And then to make your environment portable with these containers, these images, you can use different softwares. So for example, Binder. And Binder will use the documentation from GitHub and automatically build this Docker image for you. And then you can run it in the cloud and you can make it executable. Next. And now we have the part about data and code sharing. Next. So basically, you can, in many papers where there was some code executed, the authors will say that they share the data. But then when you are really interested in, or the code, and then if you are really interested in like replicating the results or trying different analyses on this data set, you try to look for those data. And then it turns out that it's actually not so easy to find either the code or the data. So what you really want to do for yourself is that people share your, people cite your work and like use your data, and we make a progress in science. So to do that, you need to share both the data and the code, and that can be used to validate your funding. It can be useful for the reviewers in case they want to try to rerun part of the analysis, and then to reproduce the results and basically build on those results as well. And then data and code might be also valuable for the, for example, policy makers, and data and code that cannot be easily regenerated is also very important to share. And then why to share, I think I already explained a little bit, but it also slowly, the culture in academia slowly changes. So the founders and publishers slowly start to demand that actually the money that was spent to generate the data or on the project, everything has to be published. And then obviously, if you made your data and your code available, some people might use it, so then you will get more citations. And then how to do that? So the best idea will be to choose the stable, open, persistent file format and create detailed documentation that the data can actually be used. And in your manuscript, include all the data citations of the source data. and finally create the metadata. Next. And what is the very common problem is that you try to, so let's say someone really shared the data or shared the code, and then you go to this website that you are really excited to see this data, and then the website is not available anymore. And that's also very common for many bioinformatics tools that they were developed, for example, for one project, and then you find the idea and the design, the code pretty useful for your analysis. You try to access it, and then the website doesn't exist. The software it's not sustained anymore. And the part of the problem is that many of the codes are developed by small research groups that are not dedicated to maintain the database or the software. They are not paid for that, so they don't improve the code. And then in the end, if someone moves to other institution, it doesn't exist anymore, unfortunately. Next. So to make sure that it will not happen to your data as well, it's even if you have your website, you can of course reference your data there, but it is highly advisable to use the repository that you can share your data with others. And at such a repository, you will need to enter many details about your data or the code. And it is very beneficial, because then the piece of the code or your data will have the identifier that you will cite in your paper. So then there will be no problem with rotten link. And not only your paper will be citable, but also the piece of data or research you produced. So the repositories are maintained. So there will be no problem with access, preservation, backup, management, versioning, and licensing. And most likely you want to have your data license. So consider using Creative Commons licenses. And then the code also, you might consider having a license for a code. And there are open source licenses such as MIT or BSD. And then how to do that, you can find some guidelines also related to open source initiative. Next. And then you can find either mandated or disciplinary repositories. So it depends on the preferences, and the funder can have the preferences about the repository. Then your institution might require different repositories. So, and it also really depends, like what kind of data you use, and you might work with like protected data. So then, for example, for human samples, there will be a different guidelines, like how to share your data. So this is quite domain discipline specific. And you can compare and find disciplinary repositories looking for repository of research data repositories. Next. And then there are general purpose repositories. So basically everybody can use them. And I think most of you will be probably already familiar with Data Dryad or Fixture or Zenodo. So these are really, really popular, I would say. For the Dryad, you need to pay. So yeah, either there's some funding from the project, or you rather use the free one, but then depending like how much data you have, you might need smaller or bigger amount of space. Next. And then, so remember about having your fair data, so findable, accessible, interoperable, reusable, and in general, get organized and be happy.
All right. Hi, everyone. I'm Aparna Shah. I'm a postdoctoral fellow in the neuroscience department at Johns Hopkins University. And you can find me at Neuromusings on Twitter. Next slide, please. So I'm going to go over the section on data visualization and analysis with you all today. But I'd like to point out that we're not really going to go into statistical analysis for today's workshop because it's a topic that actually needs an entire workshop of its own. But we did include this in the title for this subsection because we want you to at least start thinking about it and consider the importance of doing statistical analysis correctly for data reproducibility. And obviously being transparent about the techniques that you use for data analysis so that it ensures reproducibility. So data presentation or data visualization is the foundation of our collective scientific knowledge. And if you notice, figures are especially important because they often show the data for key findings. Next slide, please. So effective figures should essentially do three things. They should immediately convey the information about the study design itself. They should illustrate important findings, and they should allow the reader to critically evaluate the data that you have underlying the figures. And this last point is really important because the raw data underlying the papers are rarely available. So if the figures themselves aren't good enough, then this information can get lost forever. So I guess until data archiving becomes more widespread and is required by journals, poor data visualization practices limit our ability to interpret and critique others' findings or the author's findings. Next slide, please. So I guess Office or Sheets are frequently used for the data collection step. However, these software cannot really proxy or substitute for data visualization tools for these reasons, and I guess more than the reasons that we've stated on the slide. So reproducible workflows can be a problem with the software, even though macros and dashboards exist, because for one, a lot of people aren't familiar with these and don't use these. Secondly, Excel itself, and I believe some of you may have faced this issue. If you work with the default settings on Excel, it can often rename the genes, and that can be a big problem, especially when this data goes in supplemental figures or supplemental files, and nobody catches that error, and it goes into a publication and remains online forever. And if you need to look up examples of renamed genes, you can look up the reference that we've mentioned on this slide. But what I found surprising was that 1 in 5 or 20% of the papers in leading journals have errors in the gene names. And then lastly, the default plot options that you see with these software are often bar charts and line plots. And as we'll see in the next couple of slides, these aren't ideal formats. Next slide, please. So what you see in this illustration here is the importance of data visualization for reproducibility. And as you may have noticed, most papers present continuous data in bar graphs or line graphs, as you see on the left over here. And we really can't have a show of hands here, or maybe if you want to raise your hands in the participant Zoom window, a lot of us have used bar graphs in our paper, so you could show your hands to kind of reflect that. And I can already see a lot of hands being raised. So the point being that it is a common practice. We've all been guilty of it, but this illustration reflects why that may be a problem. So panels A through E actually are, I guess panels B through E would end up looking like the bar graph that you see in panel A. But if you actually look at the raw data that underlies these different figures, and even if you look at the summary of the statistics below, you'll notice that they actually are different in various ways. So for example, panel B is a symmetric distribution, and the data below shows t-tests either with equal variance or unequal variance, or for a non-parametric test with Wilcoxon's test. That you'd have different p-values across B through E, but yet end up with the same bar graph that you see in A. So similarly in C, you have an outlier, which you would never have noticed if you just displayed the bar graph. In D, you actually have a bimodal distribution. And if you show this as individual data points as it is done in D, instead of a bar graph, you're actually going to allow the readers to even interpret differently, or you may actually pick up something interesting in your own data. So for example, in a bimodal distribution, there could be another variable that could explain the reason the data is distributed that way. And then in E, you see that it's actually unequal sample sizes, which leads to the bar graph in A. And that can be a problem, or you should at least allow the authors to interpret the figure knowing that the sample sizes are different. So the bottom line for this slide is essentially that you want to be as transparent as possible about the data itself and allow the readers to see the raw data. And at least one way of doing that is by showing individual data points so that they can interpret it themselves and be part of the, be more actively involved in the analysis. Next slide, please. So this is a really interesting illustration that was made by Alberto Cairo, a journalist and designer, and it's called Datasaurus, and you'll see in a couple of seconds why it's called that. So the plot that you see on the left leads to the summary statistics on the right of the slide. But the designer wants to send this message that you should never trust the data based on the summary statistics alone. And there is really no alternative to showing the actual data. And let's go to the next slide to see why. So unfortunately, so this is supposed to be a GIF.
Yeah, but it's PDF, Aparna, right? Sorry, it's not working.
That's all right. So we can share this link in the chat maybe. But if you have access to the slides in view mode, you'll see that the data actually morphs into different shapes on the left in the plot, with the summary statistics remaining the same, at least up to 2 decimal points. And so if you just had one bar graph and you had some summary statistics, you would think that there's no other way to actually look at the data, whereas this Datasaurus will show that it can be a whole different way of the distributions that underlie these summary statistics. So sorry about the glitch there, but let's move on to the next slide, please. And just to re-emphasize that the importance of showing the individual data is so that the reader or the scientists who are evaluating your work, or reviewers even, can be an active participant in the study. And the interpretation itself will depend on the raw data, essentially. And as April pointed out in the chat, you can see the animation slide elsewhere. So should we proceed to the next slide, please? And so then if we've convinced you that you shouldn't just stick with bar graphs, the question then is, how do you choose the right plot for your data? So here we have 5 different types of plots, and below you'll see various criteria based on which you can decide which would be the right plot for your data. And so if you notice, it doesn't exclude bar graphs altogether. So bar graphs can still be useful for counts or proportions. So as an example, we showed you the data from the pre-workshop survey somewhere towards the beginning of our workshop, and we had actually plotted your responses as a bar graph, and that's not incorrect because we were representing counts and number of responses per, per criterion. So again, you can look at the outcome variable, whether it's continuous or proportions and counts. You can look at the sample size and the data distribution to evaluate which would be the best plot to represent your data. Next slide, please. And then again, it's not only important to pick the right kind of plot, but the ultimate goal here is to be as transparent about the data. So you want to make all the points as visible as possible. And so how does one do that? So if you look from A through E, it's gradually getting more effective at being transparent. So in the first plot on the left, the data points are actually overlapping to the extent that you can't really see all of them. So one way to solve that problem would be to decrease the point size. And then to make them even more visible, you could make them semi-transparent so the overlapping points become more disparate. And then you could introduce jitter or even symmetric jitter so that you can actually see all individual data points. Next slide, please. And then not only that, you want to ultimately emphasize the summary statistics as well, because most likely that's what you've written in the results section of your papers. And so, oh, sorry about that. You emphasize the summary statistics by increasing the width so that you can actually see each of your groups separately. And you've highlighted the summary statistics. If you see the black bar that shows the mean in this case, and then you can actually bring that out in the figure by graying out your data points so that the mean is more visible. Next slide, please. And so how can we actually create these nice looking plots? One easy solution, and it's a free option, is using these links that we have here for creating interactive plots. And the good thing here is you can actually play around quite conveniently on the bottom left in this build dot plot. If you notice, it actually gives you the option of switching from one type of plot or graph to another. And so it's really convenient to figure out which may be the best way to represent your data. Next slide, please. Thanks. And then if you want to go one step further and make them look even prettier, there are some paid options like Prism, GraphPad Prism. However, keep in mind that your university itself may have a license for the software, so you may not have to pay as an individual. And that does generate some really nice looking graphs. And again, you can, it's pretty user-friendly. The interface allows you to switch from one graph type to another pretty conveniently. And there are others like Tableau, which work with even bigger data sets, and they are nice for publications that even may go into for the public, so general audience kind of graphs. And you may have seen a lot of interesting data representation with the pandemic coverage across the globe. And so just keep an eye out and see what you think actually does a good job of representing all the data in a great way immediately, because an ideal graph sends the right message in a short amount of time without the reader having to really dig through everything. And then there's Data Wrapper and Plotly, which also are free and allow you to look at data in different formats. Next slide, please. All right, thank you.
Thank you very much. That was still a lot of good information. Okay, if you're still fit to listen, we come to the last section now, which is giving you a few tips on designing figures and images for your paper or for your thesis or whatever you can use them for. So the slides that follow are from Tracy and Helena, two of our very valued collaborators on this. And please keep an eye out, because we're currently writing a paper on exactly this, which will hopefully come out this year. It might be beginning of next year. So some of the tips, or basically all of them, in a little more context, you will find in that paper. So how do you design good figures using images? The first step is to choose a magnification and zoom that emphasizes the key message of your figure. So even if you do not do microscopy, you should really think of what do you want to show with your picture. So just like when writing paragraphs in your thesis or in a text, where one paragraph is usually one key message, think of your figure as exactly this as well. So one key message per figure with an image. So try not to confuse the reader. Aparna has hinted on that. Right now we share there's a lot of data going around right now, and a lot of pictures as well. They are shared increasingly in social media and often being put out of context also. So if you're lucky, you have a picture with a bit of a description. So specifically for that, that's not a bad intention necessarily, but specifically for that, it's good to have really one message in one figure. So this is 2 examples here. Just here, the key message is distribution of nuclei and 2 different organisms, the wild type and the mutant fly. And you see that clearly some of the nuclei are distributed differently here. So this is just one message. Second example here, you have something, again, you have one thing you want to show which is different between the wild type and the mutant. So when you have the right magnification that makes sense, then you should decide if or whether you should use color. So if you have, we go to microscopy right now, if you have an electron micrograph picture, then of course that's black and white naturally. So you don't really need to use any color. So that's would be black and white usually. If you have a picture, a photograph of something, then it's nice to have that in its natural color. If you are doing some staining or doing microscopy under a fluorescence microscope, then it's nice to use a single color, or in that case here, so that's just the example we just had. Here you could also use black and white, because here, for example, we don't really have any information. We don't add any level of information. If we are displaying the wild type and the mutant next to each other in that color that you would see under the microscope because of the filters you're using or the natural fluorescence of your sample. So you could easily make this into black and white because you actually just want to show here probably the location, or that they have, for example, here more nuclei or something like this. So black and white is a good option in that example. If you, of course, stain something, use fluorescence where you use multiple colors. For example, a very typical thing to show the colocalization of something, like of proteins or so, then it's of course good to use color. However, make sure or consider white as a color as well. And the reason why we try to emphasize to choose your color wisely is really because there are, of course, there's a considerable amount of the population worldwide that is impaired in color vision. So it's best to really use combinations of colors that are that can be seen for people with all kinds of these kind of disabilities probably. So you should avoid green-red combinations. And you can, in a lot of software, you can actually adjust for that. So this is Fiji, a software you might know. The predecessor of this was ImageJ. What works well here, so you can just see that here in the options, you have a chance to test out if it's going to work for colorblind people or not. So magenta, green, blue is a good combination, magenta, yellow, cyan, or red, cyan, yellow. And then, as I just said, use white as an option. So just to show you how this would look for people who have some impairment in seeing in color vision. Here, if you use red and green, this is not colorblind safe because people who have deuteranopia, for example, would see this as one color because they cannot distinguish between these 2 colors. So here the colorblind safe version would be here to use these colors. And then you can see what this looks like for a person who has troubles in seeing color. So this is an option, using the right colors. Or in this case, you could consider showing your channels individually in grayscale, because this also gives the same information basically. Then the next thing, if you have decided the color scheme, there are also, by the way, there's some software you can try for that. For example, Color Oracle. This is very small, it's something like a macro you can download on your computer, and then you can check if what you have on your screen is colorblind safe or not. I can post that in the Etherpad, in the shared Etherpad later on. So the next thing is to choose a labeling style that makes sense. So if you want to label structures, of course you can use lines. For example, here, then this is quickly to see. Here you're just labeling some structures, and you can use that if you can make sure that no lines are crossing over and there's just a few structures you are labeling. But as you can see, so this picture is pretty easy to interpret because you just label everything. It's written right there, and you don't have to search for any more variables or numbers. Then, of course, arrows are also an option. So arrows usually are used to help show direction or movement. Like if you have a time series, or if you say, okay, so these structures move from that point to another point. So the limitations of that is that, of course, arrows can be ambiguous. So which direction is the movement actually? And then, of course, they can obscure aspects of your figure. So you should think carefully if you want to use arrows. If you have a lot of structures to label, it makes sense to use symbols or a letter code. The thing is that the letter code should be really either written within the figure, which is possible, or like here close by, or as it's often done, written in the figure legend. However, you should always think about how easy it is for your reader to find this specific information in the figure legend. So the limitation really is that the reader needs to go back and forth between the legend and the figure, and that, of course, makes the figure a little harder to read. However, this is something that we use for teaching, for example, when we present some slides like on a big whiteboard, and then the students have plenty of time to look at that figure. So this is a good way you could use that. Then, of course, you can superimpose color code. You can use color codes on a figure to mark out specific sections of the figure. Here again, you need to be sensitive to the color blindness issue, that you use colors that are actually, it makes sense. It's also not only for people who are impaired in color vision. If you would use a red, if you have a blackish kind of figure like this one, and then you put a red arrow on it, for a person who prints out this, and people still print out papers, for a person who would print that out, or for a person who is impaired in color vision, they would not be able to detect the red on the black, because red displays as black when you have it in a black-white setting. So that's also something you should think of. So just be careful that you use it when it makes sense, and then use a minimum number of labels to convey your message. Step number 5 would be that every label, every image needs a really clearly visible and labeled scale bar, just like the one that we see here. That's like the perfect scenario when we have a somewhat sharp scale bar, and that's the scale is written right at the scale bar, and that should be big enough to read. So if you minimize that picture and it gets harder to read, that's something to consider really. Step number 6 would be to write a figure legend that can be understood, even if the figure is not longer, even if you're missing the context of that paper. So as I said, if pictures are shared online, for example, for a quick look at them, it's also good to have figures that are self-explanatory in combination with the figure legend. So people don't have to read the whole paper to get what you state in the figure. Step number 7, if available, you should really test your figure and legend with other people. So a friend from the neighboring lab, is this person able to understand the key message? Someone who's not 100% familiar probably with what you do. If this is working for them, then you designed a good figure and a good legend. Yes, I think we go back to you, April.
Great, thank you, Susie. As Susie said, we know we're together for a long time during these webinars. So hopefully some of this has, piqued your interest, and that you found at least one or two things that you can dive into and learn a little bit more about. So just to sort of wrap everything up, reproducible research practices have a lot of benefits. They help you to organize your experiments in a way that makes them more productive, more efficient. It allows you to more easily share your methods and your techniques so they're more reusable and you get more impact out of your work. It allows you to analyze your results automatically and appropriately. It allows you to share your materials so other people can actually rerun your study. It allows you to share your results more completely and more transparently. And finally, all of this together allows us to accelerate science, to do more, to share more, and get further faster. So hopefully some of the tools here have been useful for you, but we're going to get into in a moment some time for questions and comments. So if there's something here that you think that is related to reproducibility but we didn't touch upon, feel free to ask any of those questions, because all of the panelists here have a little bit of experience in different areas of this space. And when we pool our experiences as well with all the other participants, somebody in the room likely has the beginnings of an answer for you. So think about the long-term learning process and what you can do next. So what we want people to reflect on at the end of each of these webinars is just to think of one thing. So there's a lot you can do, but start with one step. Think of one step you can take to make your research more reproducible. So here on the slide, you also see our Twitter handle, our website where we share all of our materials, including the slide decks, including the handout. Let me copy the handout link here into the chat as well. All of the materials that we share are openly licensed for you to reuse as freely as you like. Just let people know about us so that they can come and join the community. This is all volunteer run, and we're all researchers ourselves. And we'd love to, if you are interested in getting more involved, we invite you to get in touch or to just take these resources and run with them and share them with your colleagues. Next. So again, we've had, over the years, little sponsorships that have really helped us take this volunteer run initiative and run with it. So we thank all of them. Next. And again, this is all Creative Commons, and you can go on our website to find all these materials to reuse for yourself or use the links that we've shared. Next. So here are all of your lovely panelists today. And we're just a few of the over 30 volunteers that have worked on this, probably many more than that now. And if you're interested, please reach out. We'd love to have you run one of these in the future. And one more slide, there is a feedback survey. So the link to that is here on the slide. It's also at the bottom of the shared notes document. So we read all of the surveys and they help us improve this webinar. So please give us your thoughts. What are your favorite sections? What were the least favorite sections? Anything like that is all very helpful. So now questions. So just a reminder that you can ask questions in multiple ways. You can put them in the Q&A, and you can also put them into the shared notes document that we've been sharing along the way. To get started, we can go over some of the ones that we've already answered as we wait for new ones to come in. Okay, so Susie, I think you shared this one. The question was, do protocol method repositories allow you to post modification? Oh no, you didn't answer that. I answered that one. Do protocol method repositories allow you to post modifications that you may employ in future publications? Or do you tend to have a distinct protocol submission tied to each publication where you're used and perhaps modified the protocol? So I started to answer that in the shared notes doc, and if any of you have experience, please share your experiences in there. But in general, yes, the protocol repositories, one of the advantage of using a repository is the ability to have versions, which allows you to track changes, but it also allows you to mark a specific version as like, this is the version that you used in an association with this specific publication, so people can always access that specific version, and then you can keep updating it so people can see those different versions over time and see when those different versions were the most recent version.
And also the thing with repositories versus putting stuff in the supplemental files. So you won't find data in supplemental files really. So if someone is searching for a data set, a specific data set. And I mean, right now during the pandemic specifically, people have sometimes more either time to dig through repositories or archives to analyze stuff that's already there because they can go to the lab. Or there's another time when, you know, these kind of searches appear, and it's really hard because there's like no automatic search button for all kind of supplements from different journals. So if you want your data sets to be available to the research community, we really strongly emphasize to use repositories because there was one of our slides said these RE3 data thing, for example, that's a search engine for all kind of repositories that are there. Even the ones that are from local universities, like my university is a technical university, we have our own repository for our university stuff, and even this one, you can search through with these very open meta crawler kind of research sites. So really that's the thing, if you want your data to be found, to really use the repositories. That's another thing I think that speaks for them.
So the next question that we have in here that was, Susie started answering this one. It says, regarding file organization and raw data, do you suggest when the raw data are used in multiple major distinct projects, or what do you suggest when the raw data are used in multiple major distinct projects? Do you copy the raw data into a raw data folder in each project so there are duplicate copies? Do you go outside of the project folder to one main raw data folder that all projects rely on, or something else? Susie, do you want to start on that one?
Yeah, so that's because I'm working with another lab in a different country, and what we do is we even use Excel files to share them in between computers. The bottom line here is, as long as your raw data are in a format that allows you to easily share them with others without they having to use a specific software, then this is, of course, that's what you should do in general. And of course, you make copies of that. So when I said you should leave your raw data pretty much untouched, what I meant was specifically, do not, for example, have your raw data in an Excel spreadsheet on one sheet, and then on the next sheet you do your analysis in them. Because sometimes, because of dependencies, when you use a certain formula or something, sometimes these things can get changed and you have no way of knowing that your raw data are not the same as they have been from the day that you got them. So this is why I said you should keep them kind of untouched. Of course, you can make multiple copies of them. And I like the discussion that was going on. So someone said, yeah, but if I already know I have like an error in there because it like should be 25 instead of 2.5 or something, of course you can correct the mistakes in that. And then someone else jumped in and said, well, I always leave it untouched even if it has some errors because I just use code to correct for that. So I like that approach to really people use different ways, and it's also really nice to know that people think of this differently. So again, it's not one size fits all. The bottom line is really you should try to protect your raw data from getting changed accidentally. And we hinted on that Aparna said that Excel tends to rename things. So because Excel is not really what you see is what you get kind of thing. So it can happen without you noticing it, and that's a bad thing. If you then 2 years later go back to your raw data and then you realize, oh my God, there's a mistake in here. I didn't see it, and then it blows up your whole analysis, and you have to do it again. And it takes another half a year or something. So yeah, sure, share them and make copies of them. Sometimes raw data are, for example, I work with people that do GCMS or something, or they have very specific analysis. So they do like analytics, where you kind of define what kind of compounds you have in a sample. And often these softwares are arbitrary, or you need a license to have them. So I can never access these spectra, for example. So I need someone to convert that for me into some sort of readable format, so I can have access to that. So that's the thing with which format should I store it in. I have just one more kind of tip for that. So if you can, you should use the format to store your data in should be a format that has probably the best chance to survive the next 10 or more years. So to probably be a format, that there's LibreOffice or OpenOffice, which are right now run in parallel. So it should be preferably a format that can be read later on as well. Or if you have some text, you have a chance to convert it into PDF or so, because PDF is pretty much unchangeable. So this is something you could try, for example. This is just 2 more cents on raw data. I hope that helps.
Awesome. So the next question that people started answering in the notes was, why or when would the date be useful or needed when you can see that in the metadata of any file? So this has to do with file naming. So we have 2 answers already in the shared notes document, which is helpful. One is that sometimes those dates in the metadata can be corrupted or changed, especially when files are passed back and forth in different operating systems, uploaded and downloaded, whereas in the name, that might be less likely to happen. The other thing is that how you decide to create these little informative sections of your name can also help you to filter files. So especially if you're creating many, many, many files over many years, by having a standard name in your file name, you can filter by day, you can filter by month, and you can filter by year. For some types of projects, that can be very helpful. So it does depend a little bit on what your needs are, but those are some of the reasons why some people will recommend putting the date in the file name itself. And again, to reiterate, it's not one size fits all. So that's what some people choose to do, and it helps those particular cases. Does anyone have anything to add about any of those first 3 questions? Any of the other panelists?
I have just quickly follow up on what you just said. So I experienced when you use Windows, for example, and you switch, like you update to Windows 10 or something, and then you go back to another machine. Sometimes the dates don't match, or the actual the time, so it says like, okay, this you recorded this picture at 3 p.m. and then you open it in another machine, it says, oh no, that was 4 p.m. So so really Windows does that as well. You will have no chance of knowing. You just start it in a confusion because your 100 files are all 3 p.m. and the other exact same thing. Windows says, oh no, it's at 4 p.m. So do yourself a favor and just I usually tell my students, rather be a bit redundant. Just do it. Don't think about it. Just do it. You will thank yourself in half a year or something, because you are your most likely collaborator of the future. In half a year, you will not know what you did today, likely. So it saves yourself a lot of sanity. And even if it's kind of in vain and you did it as an extra thing, I really found this is super helpful. So rather be a little bit redundant. Probably that helps as a mindset as well.
Awesome. So we have 2 new questions in the Q&A. One is a question about versioning. So they've been using Git for 6 years and during the semester on almost a daily basis, but like many people, they don't understand it and find it difficult to use. This is very common. It's especially difficult when they don't use the same routines, when you don't code frequently, There are a lot of steps that people can take. Oh, are there sorts of steps that people can take to one day be super proficient with versioning, or what is the best way to find versioning for you? So I'm going to just start with my opinion on this because I have a strong opinion about it. And then we'll pass it back because I know that a lot of the other panelists have similar experiences and may have a different take on it. But for me, if you are what I call an infrequent programmer, which most researchers are, then trying to adopt Git may be a huge challenge. And what you may want to adopt are tools that have versioning built into them already. So it's definitely commendable to try and use Git, but some of the electronic lab notebooks we discuss, such as the Open Science Framework, have built-in version control already, so you don't need to learn how to use Git. If you're using R, RStudio is integrated with Git, so you can use Git through RStudio without using the command line. It's using this command line, the terminal, that many infrequent programmers have a challenge with. So see if you can find some tools that have versioning built in, and then that can really improve your documentation with a less of a steep learning curve. So I welcome anyone else to jump in on their perspectives on that, the difficulty with adopting Git as a versioning system.
I think I would just add, I guess, resonate with what April just said about a lot of us not being very proficient as coders necessarily, but how electronic notebooks can really help with that. A digital trail is certainly helpful, and in itself can be a way of controlling for different versions. So I don't have anything new to add, but I agree that I've felt the same way myself as a researcher.
I mean, if we talk about versioning also for like manuscripts or something, what we do nowadays is when we write a manuscript together with like 3 or 4 people, you will have your changes, like you may have some track changes, you use Word or something, and then you will add like with an underscore the person who was last working on it, and then it's like version 6 or so, and then the next one gets it, and then you delete like the person working on it before, but then you have your own like initials, and then it's version 7 and that kind of stuff. So this is versioning you do manually, of course, but this helps with a manuscript to really know what version are we in right now, because otherwise it just gets confusing if people don't or cannot use something like Google Docs or so, which can of course pretty easily be used with writing a paper together or so. So, and the second thing is with versioning. So again, so if you have, for example, we do a lot of qPCR, real-time PCR, and I usually, when my students do something like 3 PCR each day, I just let them also, again, have the version on it, and then I always know exactly, okay, so this is the date, this was the person doing it, and then when I get back to that half a year later, I know, okay, that was the person, and I can just sort it in my head. So it's, and for them, it's helpful to know that was the first PCR I did today, so I was still fresh. And the third one I can throw away because like my mind wasn't, I did a lot of mistakes and pipetting errors or something. So that's also good to know then, okay, so that was the third 1 hour pipetting thing of the day in the evening at 5 or so. So maybe I shouldn't trust that so much. So again, that's some manual versioning thing, but that's nothing really to do with programming, of course.
And we have a suggestion in the shared notes from one of the participants that say that they make cheat sheets for Git that they use to refer back to. And I bet you even you can find some public ones. I know that R has some great cheat sheets, so I'm sure that Git has some too. So that's another way if you are wanting to adopt R, that's a good way of learning. So we have one more question in the Q&A box. Do you recommend to also include the code on how figures were generated, for example, R graphs? So everybody's nodding their head, and that's because that is definitely one of the missing pieces when we talk about a barrier to that baseline of reproducibility, of generating the same results from the data and the code. So someone want to jump in and talk about that a little more?
So Gosha and me, we've been working on that because when we were co-writing that paper. So this is really, that would be the cherry on top, really. So if you can include that, and even if you feel like people right now are not doing it, just do it. You will set a new standard for the field. And the more people that actually do that and really report how they created the figure in a very transparent way, you will be one of the leading figures then, probably at some point. And I have another tip for those of you who are young researchers, and when, for example, you write a manuscript with a PI or an older person and they are not supportive of you describing everything in detail, what you could do to circumvent them and still have your work shine in a way is you could deposit your protocol, your specific protocol with protocol.io, for example, and then link to that in the manuscripts method section. So you have a very clean, nicely written description of what you did, or you use GitHub for that. So a very lengthy section so people can really use that. And still, so your work is still attributable. It has a DOI, it's citable. So this is another workaround if you work together with people who right now do not support probably to report all this because they are not used to it, or they are currently, or maybe the journal says, okay, we want the method section to be shorter, this is too long, this is not our standard. You can get around this by depositing that protocol or something on another place and then citing that. And this is, it's not hard to do this. So protocols.io has a great support right now thing going on if you want to deposit your stuff there. So there are also various other websites who do that. There's bioprotocols as well. So give it a try. It's easy to use. It's much easier to use maybe than GitHub because you basically just have that chunk of text you put there and then you check if everything's correct and you click submit and then that's done. So please do that. Please put the code to your figures there. It really, people will thank you later and you will be cited more often actually. So we saw that if you're very transparent in reporting, people will cite you more often because they're also grateful for the work. So it's not forgotten or anything.
Yeah, and so go ahead, Aparna.
I was just going to emphasize one thing that Susie brought up, which is the limits that manuscripts have, or journals have. Don't let that be an excuse for sharing anything. So we're working towards, or an ideal situation would be where journals just don't have limits at all, especially as they transition to being digital and online. But until everything is online and there are no limits, we can't use that as an excuse, because just as we've seen through this workshop, there are a lot of ways to just put everything online and link to it. So yeah.
Yeah, that's an excellent point. And just to sort of add a little bit on that, one common example for sharing code that's free, especially if you already are using GitHub as a place to collaborate on code, GitHub is integrated with a free repository, I believe it's supported by CERN, called Zenodo. So that integration makes it very easy to do a release of your code that supports those figures in your paper. You get a DOI for it, so people will be prompted to cite it. And so that's all free to use. So in the last few moments, I'm just going to share in our chat a link to our post-webinar survey again. So it's very helpful to get feedback from everyone so that we can continue to improve the content and how we do these workshops. Does anyone have any panelists have last comments, things I may have forgotten to say?
I was just going to add, and I was just typing it in the chat, that if you have questions in the future or even in the near future, you can even tweet at the Repro for Everyone Twitter handle and use the hashtag, which I believe is also Repro for Everyone. Again, like April mentioned, it's a collaborative team of people who have worked in various fields and all over the world. So hopefully someone will have an answer, and Twitter is a great place to find those answers.
Thank you all so much for being here. Thank you to all of our attendees and all of our panelists. We really, really appreciate you taking this time to share this information with us. I'm already thinking about how I can reorganize my data. And this is the last event of ECR Squared. Most of us are starting our semester right now, so good luck in the semester to everybody, and thank you for joining us for all of these events. And I also really want to thank our moderators that helped us out here today and through all of our events, and then the grad students from ASN, SSE, and SSB who all really helped put on this event, along with the leadership from these three societies for being so supportive. So thank you so much for spending time with us today and for spending time with the whole evolutionary biology community throughout these events. And I hope that you're able to take some of what you learned today and incorporate it into your research. Thank you so much, everyone.
Thank you. Bye.
Have a nice day, evening, night.
Depending on the time zone.
Rest of the week. Have a nice rest of the week.
Thank you.
Okay.